Red blood cells carry hemoglobin and circulate throughout the body to supply oxygen. Beta-hemoglobinopathies are blood diseases affecting the structure and function of adult hemoglobin. These genetic disorders are caused by mutations in HBB, the gene encoding the beta-globin subunit of hemoglobin. HBD is homologous to HBB, but with much lower expression due to a weak promoter. We asked if genome editing of the HBD promoter could enhance adult hemoglobin levels. Using CRISPR-Cas9, we inserted transcription factor binding sequences into the endogenous HBD promoter, boosting its transcriptional activity and leading to increased HBD expression. In bone marrow stem cells, elevated HBD levels generated delta-globin subunits that assembled into functional hemoglobin complexes. Our work is a promising approach for restoring hemoglobin levels in red blood cells. This strategy might open new therapeutic avenues to treat beta-hemoglobinopathies in the future.